Hello, today, we will understand the physiology of proximal tubule. First, you should know about molality and osmolality. Molality is the moles of solute per kilogram of solvent, which has been used in chemistry. While osmolality is the concentration of dissolved particles in blood or urine. It is defined as milliosmoles per 1 kilogram of solvent. Osmolality in glomerular capillaries is 300 milliosmoles per kilogram, and filtrate coming from Bowman's capsule to proximal tubule has same osmolality of 300 milliosmoles. So, filtrate coming in PCT contains solute like sodium, potassium, calcium, magnesium, water, glucose, amino acid, lipids and small proteins including creatinine. In proximal tubule, filtrate undergo two processes, tubular reabsorption and tubular secretion. Let's first see tubular reabsorption. Tubular reabsorption is a process by which various substances present in filtrate are reabsorbed back into blood from lumen of the tubule. This is done with the help of epithelial cells in proximal tubule. Here is the PCT epithelial cell. This is blood vessel. Surface of cell which is facing the blood is the basolateral membrane, and the surface which is facing lumen is the apical membrane. Here, absorption occurs through channels present on epithelial cell. The first and most important is sodium-potassium ATP channel at the basolateral membrane, that pump 3 sodium outside, and 2 potassium inside cell against their concentration gradient that lead to decreased sodium level and increased potassium level in cell, initiating the gradient by the which sodium will move from high concentration in the filtrate toward low concentration in cell at apical membrane. Now this sodium will also bring other substance with it, acting for them as a source of energy like glucose, amino acid, lactate, and chloride ion. This is an example of secondary active transport. The reason is that these substances are moving against their high concentration present inside cell. Carbon dioxide diffuses into the cell and combine with water and form carbonic acid in the presence of enzyme carbonic anhydrase. Carbonic acid will break down into hydrogen ions and bicarbonate ions. Hydrogen ion will be transported through sodium and hydrogen antiporter in the lumen. Hydrogen ion combine with bicarbonate ions present in filtrate and form carbonic acid, which break down in the presence of same enzyme carbonic anhydrase present in the cell membrane into carbon dioxide and water. Carbon dioxide and water will absorbed into cell. 90% bicarbonate will be reabsorbed. 65% of sodium is absorbed in this area, as water tends to follow sodium. 65% water will be reabsorbed. 100% of glucose, amino acid and other solute are reabsorbed. Magnesium, potassium, calcium are absorbed through the paracellular spaces between cells. Lipids diffuse passively through the phospholipid bilayer. Small proteins like insulin, hemoglobin are endocytosed by the cell, where they combine with lysosomes, digested into amino acids which are absorbed to blood. Phosphate ions are reabsorbed with help of sodium, in the same way like it helped to absorb glucose. But phosphate ion absorption is regulated by parathyroid hormone. Parathyroid hormone bind to receptors present on the cell. This activate adenyl cyclase, which convert ATP to cyclic AMP. CAMP activate P kinases A, that phosphorylates sodium phosphate ion channel. Inhibit this channel, due to which phosphate ions will excreted in urine. Parathyroid excrete phosphate ions to prevent its crystal formation with calcium. Tubular secretion is the process by which various substances are excreted from the blood back into the lumen of proximal tubule. This includes secretion of phosphate ions as we have discussed earlier. Hydrogen ions are secreted by sodium-hydrogen antiport. There are two energy-dependent transporter. First one, organic anion transporters that transport weak acidic drugs like salicylate, methotrexate, penicillin, and uric acids, and second, organic cations transporters that transports morphine, dopamine, and creatinine. Ammonia and bicarbonate is produced from glutamine. Ammonia can exist in two forms, as ammonia, which is neutral, or ammonium, which is positively charged. Ammonia freely diffuses from PCT cell to tubular lumen. Here ammonia bind with hydrogen ion to form ammonium, which gets trapped in lumen. While ammonium substitute hydrogen ion in sodium hydrogen antiport enters tubular lumen, get excreted in urine. Bicarbonate got to blood in exchange of chloride ions.